We're going over my notes about. Oh, Hi, peeps. We're continuing with my cell. Okay. About we're doing cell biology and life science in a nutshell. We were at the section of learning about the cell membrane, going over some details. This is the glycerophospholipid molecule, which consists which well, it consists of a hydrophilic tail, which we learned in the previous video, and hydrophilic head. The hydrophilic head consists of three sections: a choline in the blue, phosphate in the orange, and a glycerol in the green. So choline, phosphate, and glycerol, and that's um, the tail, which is fatty acids. So that's what it is. And then again, the glycerophospholipids are far the most abundant lipids in cell membranes. Like all lipids, they're insoluble in water. Their unique geometry causes them to aggregate into bilayers without any energy input. This is because they are two-faced with hydrophilic or water-loving phosphate heads and hydrophobic water-fearing hydrocarbon tails of fatty acids. In water, the molecules spontaneously align with heads facing outward, the hydrophilic, and tails lining up in the bilayer's interior, the hydrophobic. Thus, the hydrophilic heads of the glycerophospholipids in the cell's plasma membrane face the water-based cytoplasm and the exterior of the cell. So you have two molecules making up the bilayer. So the two fatty acid tails face one another, and the two hydrophilic heads face on either the cytoplasm or the exterior of the cell. So it's called a bilayer because there's two glycerophospholipid molecules that make them up. Altogether, lipids account for about half the mass of cell membranes. Cholesterol molecules, although less abundant than glycerophospholipids, account for about 20% of the lipids in animal cell plasma membranes. However, cholesterol is not present in bacterial membranes or mitochondrial membranes. Also, cholesterol helps regulate the stiffness of membranes. So that's showing the bilayer with the hydrophilic heads in green round. The purple are the tails, which face one another. So that's what the bilayer is looking like. And a double bond over here on one of the fatty acid tails causes a kink or an asymmetry in the tail. But when they align. They're all the same shape. So then, so we have cholesterol is also part of the bilayer, regulating the stiffness of the membranes. And then also other less prominent lipids play roles in cell signaling and cell recognition as well. Another picture showing the bilayer. In addition to lipids, membranes are loaded with proteins. In fact, proteins account for roughly half the mass of most cellular membranes. Most of these proteins are embedded in the membrane and stick out on both sides. They're called transmembrane proteins. The portions of these proteins that are nested amid the hydrocarbon tails have hydrophobic surface characteristics, and the parts sticking out are towards the outside, and the cytoplasm are hydrophilic. So that's showing that. At physiological temperature, cell membranes are fluid, or meaning they move around. They're not perfectly stiff and rigid. At cooler temperatures, they become more gel-like and a little more rigid. Scientists who model membrane structures and dynamics describe the membrane as the fluid mosaic in which transmembrane proteins move laterally in the lipid bilayer. Therefore, the collection of lipids and proteins make up a cellular membrane, relying on the natural biophysical properties that control the form and function of the membrane, and as well as the temperature, etc. In living cells, however, many proteins are not free to move. They are often anchored and placed within the membrane by tethers to proteins outside the cell or the cytoskeletal elements inside the cell, or both. Cell membranes serve as barriers and gatekeepers. They're semi-permeable, meaning that some molecules can diffuse across the lipid bilayer but others cannot. Small hydrophobic molecules and gases like oxygen and carbon dioxide cross membranes rapidly. Small polar molecules, such as water and ethanol, can pass through membranes also, but they do so more slowly. On the other hand, cell membranes restrict diffusion of highly charged molecules such as ions and large molecules such as sugars and amino acids. The passage of these molecules on specific transport proteins embedded in the membrane is called selective transport. Specialized proteins in the cell membrane regulate the concentration of specific molecules inside the cell. So we did review this in earlier videos as well, but just going over it. You have different channel proteins and membrane proteins, transport proteins. This one's a glucose transporter, transporting glucose into the cell and whatnot and outside. These are channel proteins moving ions across channels into and out. So depending on the concentration gradients, these move, whether these molecules are needed inside or outside the cell. And here's showing amino acid movement as well across membranes. Membrane transport proteins are specific and selective for molecules they move. Often they use energy to catalyze this passage. These proteins transport some nutrients against the concentration gradient, requiring additional energy, or ATP, or using GTP, etc. The ability to maintain concentration gradients and sometimes move materials against them is vital to cell health and maintenance, because one molecule may be more needed in one place rather than another place. Thanks to membrane barriers and transport proteins, the cell can accumulate nutrients in higher concentrations than exist in the environment, and conversely dispose of unneeded products as well, depending on where molecules are needed the most at different times in the cell cycle. Other transmembrane proteins have communication-related jobs. Proteins bind signals such as hormones or immune mediators to their extracellular portions. Binding causes a conformational change in the protein, transmitting a signal to intracellular or messenger molecules inside the cell. Like transport proteins, receptor proteins are specific and selective for molecules they bind. The action of transmembrane proteins or transporters carrying molecules such as glucose from one side of the plasma membrane to another, or receptors binding extracellular molecules and activating intracellular process. Enzymes also. So that's how they work, transforming molecules into other forms. And anchor proteins physically link intracellular structures with extracellular structures. So transporters, receptors, enzyme, and anchors, different kinds of membrane proteins. That's shown again the phospholipid bilayer, and so the proteins are anchored inside the bilayer. So we'll continue, and peripheral membrane proteins associate with the membrane externally. See you in the next video.